Để mở video cho thầy đỡ cô đơn thế này. Tụi nó đóng video hết rồi. Không nghe thầy nói gì thầy ơi.
Thầy unmute đi thầy ơi.
Nghe xíu xíu. Nhỏ lắm thầy ơi. Nhỏ lắm thầy ơi. Nhỏ xíu luôn thầy ơi.
Hello.
Thôi thầy ơi.
Okay, good. So we have some people today, not much. Oh. Okay, so thank you very much for joining me today. It's kind of hard time. So anyone okay, right? You have this one, right? You have this one, right?
Yeah.
A minute. See it. Wait a minute. I need to familiar with this one a little bit. Hello, everyone. So, yeah. So sorry so much because of the. I thought the staff from the IT service center already sent you email for today, for the meeting today, but maybe you didn't notice the email from our university email. So it's okay. So let's check is how many people around us today. Quite a maybe more than 20 people. That's it's okay too. It's a long time to see. And next thing I want to show you is the lecture. Kind of we missed some lecture. I think this week is maybe in week number 5 or number 6 already, but we just have the 2 or 3 lecture, and we kind of a little bit late because of me. Sorry. And I plan to speed up a little bit and also modify the text for the midterm. And anyone can, before that, I also have the one day for review. This would be so hot for the contents you need to grab for the midterm. So today is kind of important, but also very basic. I think most of you is familiar, not 100%, but many of them. So I will go. I just hear the voice from Nam. So can I hear from voice from another people for not feeling lonely here? Okay, maybe you are kind of busy some stuff. So anyway, I will start the lesson today. We mainly talking about the main stuff of our course. That is genetic variation. Is the first part. We'll talk about origin of the mutation, and I try to remind a little bit about the term mutation in the disease. So the mutation is simple, right? The change in the nucleotide sequence of DNA is a mutation. It's simple. But like the first lesson, we discussed shortly about that. So the change in the nucleotide sequence, we have many levels. So, sorry, can I ask questions? So can you see the slide, right?
Yes.
Okay. And the change in nucleotide sequence is quite diverse from a small scale of the single nucleotide chains, second nucleotide chains, or we have the more than 1,000 or a megabase chain of DNA or whole chromosome chains. But sometimes, some case, not in human, but another species, we have the chains whole genome. Whole genome is a kind of chain number compared with a normal case. And a second term we need to follow is a disease. Is a, like you see in text in here, is a particular abnormal condition in your body have the negative effect. And they affect the structure and the function in part or all of your body. So it's not due to any external injury. So you can see that in the picture I'm using for illustrate that, you see these we all human, we own same species, but we see it's kind of the skin is different, right? The hair color different, and the face is also different, and a lot of things inside also very different. I mean, it's the cells biochemical reaction, or it's the ability of the enzyme and amounts of enzyme or amounts of the chemical in your cells or your tissue also different. So that kind of thing is subtle. It's not showing out in your field outside, external outside. But I had a question here. So maybe you, based on the basic in here, you may have the question. So how are the people got injury and had to change the kidney? And then because of that kidney is not belong to his cell or her cell, so the kidney and it, is the immune system in the immune system of our body is not accept the new part enter the body, so they try to find into new organ, and the new organ also have the immune cells, and they fight back to the normal cells next to the new environment. So in this case, can we call it disease or not? And for fast, the answer is yes. That's also one disease, because the new part is also your body now. So because of the abnormal in the new kidney, so this we call it also the new disease. So in that case, we can that type of disease also what is this? Famous. We call this because that one is graft versus host disease, GVHD disease. So this also make the people is receive the organ, new organ is kind of to suffer a lot. Okay. So what a factor can be contribute to the phenotype variation? So we all know is the phenotype is a sum of the effect from the cause is phenotype, the genotype, your gene, and interact with the environment. So if the environment is not different between two people, but two people is different, so we can see that all the difference between two people is from gene. So the difference between two people from gene and also interact with environment may be very large. In here, you can see that the very tall people and very short people, it much different, maybe 3 times or 4 times different in height. And they also is different in the skin here, and also the strength and behavior, good or not good also. So that's, you know, is based on the gene or environment, like we all know about that. So what is so hard. So we have the now we come to the mutation. So you know, the mutation is a change in the gene, and they may have effect into your phenotype. But don't think it's all of the mutation will have impact to your phenotype, because of a lot you know is we have between the 2 people we have more than 5, I guess 5 millions of the nucleotide different. Not all of them will contribute to the difference between the 2 people or contribute to the disease. I mean that not all of the mutation have the impact. Just small scales, small amounts of the small number of the mutation have impact to your health status. So we can divide the mutation type by origin or by cause. So origin, we have the germline mutation. So I just remind again. So germline mutation, that means from the zygote, the sperm of your dad and the egg of your mom is make the zygote. From the zygote state, you already have that mutation. And germline mutation, I mean all of your cells have that mutation. Any cell in your body also have that kind of DNA code. And that mutation, how about the next generation? The germline mutation from you also can transmit to next generation. So we have the link between the grandson and the grandfather. And another also very important, and right now in the disease outcome, we really need to remind them, is a somatic mutation. The somatic mutation is acquired through the living time at some cells. I mean, it's not 100%, but few % of your cells have that mutation, maybe in your skin or your lung or your eyes or your brain or something. So, and that mutation, the question is that mutation can transfer to next generation or not. Of course, that mutation is not from your mom or your dad, not. But the question is, they can transfer to your son or your daughter or not. No, they cannot transfer. But also, yes, if that somatic mutation happen in your germline stem cell. So you know, is when you were baby, so some cell is already received a signal to become a germline stem cell inside of your body. So if you have the mutation in there, so all of your sperm or some part of your sperm also have that mutation, and they can transfer that mutation to the next generation. So, and of course, they have it. Few of the germline mutation, because they also have the application when a small population of germline stem cell to become a large germline stem cell, because you know we have a lot of sperm and a lot of egg in the lifetime. So some of somatic mutation also create and transfer to next generation, but I think few of them, and we have the probability of that, but few of them can transfer to next generation. So that is quite a very common question if you discuss about this topic. And the somatic mutation also rely with very hot topic, like you know, it's cancer, right? Cancer, because of maybe the environment, so they induce some mutation in your body. So they create somatic mutation and somehow they develop to become cancer. And the second thing is by course we have the two things by the spontaneous mutation. That means naturally happen spontaneous mutation, naturally happen by cell duplication. And this, you know, that is we have 3 billion base pair, and then when we make the photocopy and some character, I mean, some DNA code will be changed. Actually, we have some kind of mechanism to fix that kind of wrong copy, but somehow is we cannot fix all of them. And the second is by course is we have induced mutation caused by the environment agent. That means we living in the harmful environment, maybe the pollution, or we drink so much some toxics, some sugar a lot, or we smoke a lot, drink a lot, or some unhealthy thing. Okay, so that's quite very basic about the mutation type. And a next question I want to check from you is here. So how to check the mutation? How, for example, if you take, for example, you have some tumor. I don't know if that tumor is the cancer or a kind of a friendly tumor. But if you have that tumor and you like you know about some molecular technology and you want to isolate DNA from that, you take a little bit small of the biocide and take a little bit meat from that skin and you sequence that. You found some mutation. Compare with the reference genome in the internet, and you found some mutation, and you don't know if this mutation is germline mutation or somatic mutation. So how can you check? How can you check that is germline mutation or somatic mutation? If you're somatic mutation, you may a little bit worried because they can develop a cancer, or a germline mutation maybe also safer a little bit because transfer from the mom. So somebody can answer the question. How can I see your question? You can type it or just discussion. That's also interesting. Nghĩ. Can you hear me? Hello. How can we check the mutation that is belong to somatic mutation or the germline mutation? We kind of discussed a little bit. Now, say something.
Yeah, I just type on the chat box and I say like test the parent and like the parent genome like Tom say in there.
Where chat box? I don't know. Here. Oh, chat. Okay. Test parent. Compare with parent genome. Test parents. What if that people is what if that people have no parents? I mean, if parents pass away. So, and also take the DNA from the parents is also a little bit trouble, right? Because of the you have to maybe live in another country how to take that, right, Tom? So quite simple, right? Because of you have a tumor and you think that it's somatic mutation, but I mean, the somatic mutation just maybe less than 1% of your body, maybe very less than 1.1%, very small. You can take another DNA from another side of your body to compare, and then you can identify that that is a real somatic or a germline, because of 99% of your cells is a germline, and just small part of that is a what somatic. Why I said that we can compare with tumor and normal people. You can say that is that we can compare tumor and normal tissue, not the people, because the two people we have a lot of mutation, a lot of the SNP. You cannot compare like that one, right? So compare between the diseased people and the normal people is have a meaning when you want to identify that the mutation is harmful or not. That is that's mutation, a good mutation or a bad mutation. You can compare with normal people and illness people. But if you will compare, answer the question like germline or somatic, you can simply just sequence another side of your body and compare, right? Being said that, I need to turn on the volume, but I need to maximize from mine, and I look like another people have no problem with the voice yet. So check your side, okay? Okay. Okay, I'm not going to, just a slide number 4, because we take a lot of time. And this slide just remind you something about the term like, it's better if I can write something in the board, it's better, but no. So you know it's locus and loci, locus, right? Locus. Normally we not just focus on the gene, right? So somebody is think it's locus is just talking about the gene body is not correct. So locus is some part of the genome and genome in the comparison between the another people in the same position in the genome. That's locus. And maybe they also talk about the location of the mutation. Yeah, and loci is a separate locus will make the loci. And the polymorphism is we already know about polymorphism, not the polymorphism. And the next is the alleles. So alleles is the varieties of the gene. I mean, it's not like we think that gene body, like we have ATG or a TAA from the start to the end, not the locus. So just part of their genome. And what's alleles? So alleles is the different form of that locus. So we have just single chains in that locus. Also, we have the one alleles, and another in the next sister chromosome is we have another alleles. If that people is a homozygote, heterozygote, and homozygote is we have the same what we have the same DNA code in that position, and heterozygote we have it different. And genotype is we talk about that people have the diploid, right? From we have one chromosome from that, one chromosome from a set of chromosomes from mom, and that is sum of that position we have a genotype. And how about haplotype? Maybe a lot of people is not familiar with haplotype, but haplotype is quite very important. So if genotype we just focus on maybe single nucleotide, single nucleotide, and we can say that one allele or two alleles. And genotype also considered for one single nucleotide in that single base pair. But haplotype, at least we have two. In the haplotype, at least we have the two base pair together to become a haplotype. That means that's two mutation. They need to go together to next generation to become the haplotype. So haplotype, that means both S and P to belong to one chromosome. So we know this haplotype. If they belong to different chromosomes, they're not haplotype. They belong to different haplotype. Any question from this? If I can roll something, is better for. Okay. This one is Punnett square. So you maybe very easy to got this one because the same with the dominant and recessive in the experiment of Gregor Mendel. So we had a parent with log H and log H, and another parent we had a small lowercase here. So 100% we have that case. Okay, and this 100% we have the heterozygote. And if the parent is of both a carrier or heterozygote, so we have the ratio is 3 per 1 for the phenotype. That is the case of dominant and recessive. But in normally, dominant and recessive is not always correct because we also have the one case is a co-dominant. That means two types of alleles also have a meaning to the phenotype. For example, what is a sample? What is a good example for this? Like you see that this also is a blood type, for example. Blood type. So mom is blood type AA, and dad is blood type is BB, and the son or daughter should be the A and B, and they are same function to the blood. And then another is a Mendel experiment. We have the flower color of the or a cell receptor. They also different type of the gene. Different type of alleles. So we have a little bit different in the type of receptor. For example, in the MHC receptor, they can recognize different case of the bacteria or viruses enter in the cell for immune system reaction. And also in human skin, like Barack Obama, the president of the USA, also the hybrid from black father and white mom. So human skin also is not dominant, there is a co-dominant. So for easy to illustrate that like this, okay, this is co-dominant. So both kind of the characteristics show in the baby, right? So perfectly fit. And also for funny is this is not, right? This is a baby don't have any kind of the stuff related with the mom or dad. I mean from the skin. They didn't transfer to the baby, just for fun. Okay, next we, so a previous slide, we talked about the type mutation related with the cause, related with the origin, and now we talk about the type mutation related with the scale. So we have the genotype duplication. So the duplication, and one is a genotype, one body is a genotype duplication, and one body is not duplication. They're different, right? But different is too much. That means six, what, three, 3 billion base pair. The amounts of DNA genetic material is very big. And we have the chromosome level mutation. I mean, if the number of chromosomes may be changed, but not whole genome. Number of chromosome mutation, and also part large scale means the chromosome segment is different between people to people. And another one is gene mutation. I mean, a small amounts of genetic mutation is different. Maybe that's a single or some hundreds base pair from, or some thousand base pair. So, so genome duplication is very popular in the plant. For example, some kind of banana is 3N. And this definition is we have an additional copy of the entire genome. And due to this mechanism, due to a sequestration not equal during the meiosis, not during meiosis. So one gamete have no any kind of chromosome, and one gamete have the 2N. And you can see that that's gamete to make the new zygotes, and you have the 3N or 4N. So the result is a polyploid. In the plant, we have a lot, but in human, I don't know, we have it or not. Maybe we have, but they quickly die, okay, before get birth. Gene duplication is an important factor for evolution. So I also mentioned several times already. So I mentioned several times already. So the very first creature in our planet is the genome is very small. So that small is very less material for doing some work. So they need to increase that. And the most effective way is genome duplication. So it's here, we have the Euro called that, they have the ancestral gene is very small, and then they underwent their genome duplication. We have two kind of copy of gene, and if we can detect in the fish like this. So in this species we have a haploid, and this species we have a diploid, and number of genes is increased. And in case of the go to higher evolution, along a later evolution, so this next gene is they underwent some kind of the gene duplication. I mean the small scale duplication, not whole genome anymore, but that gene is somehow they multiply, they jump by their homologous chain, like in the cells class we discussed. So this one will be increase the number of genes. But the good thing is you can see that the functional gene, the first function of the first version is the chain. They have a very similar in DNA sequence with the first gene, but the function is just chain to adapt with the more complex need of the new host. I mean gene in the host. The one is one also interesting question, and you will met later, is we have more DNA, more material DNA, and sometimes we lost the DNA. So which one is better? Which one is better? So you can see in the plant, I just type it from the internet. So this is a polyploid. This is a diploid, normal, and this is an abnormal with a genome duplication. You see that it's bigger a little bit. And the bigger floor plan, that's also good for them because of the human and also the animal try to eat this one bigger. Maybe the quality may be a little bit delicious. And then it's good for this kind of plant can transfer the good stuff to the next generation. So that is the one I need to mention to you is in the cell biology or also in the genetic. So more is better than less. So if you have more copy of gene, I mean the amount of protein made from that gene is larger compared with the normal case, and that maybe abnormal, maybe that is a disease, but it's better than less. If you less 50% or less, if you are decrease about 80% of the amount of protein produced that work, that's not good. They will die soon, and you can see that later. Hello, our connection is still okay, right?
Yes.
Not sleep, right?
Yeah.
And can you hear your voice? I hear your voice.
Yes.
Okay, that's great for me to continue that. The next thing, we talk about the chromosome mutation is quite a, I will go fast. This thing is because you already know about that. So we had a change in the structure of chromosome, loss or gain part of chromosome, or very tiny part of chromosome change by another code. So chromosome mutation, we have a lot, like not a lot, just a 5, or some kind of more diverse, more complex. It didn't go here because of you know, it's in this field about medical genetics. So a lot of fighting more and more to see the different and try to explain the disease outcome in the people. So this new finding also update day by day. So very much information. So we have the chromosome deletion, segment deletion. So in here we lost a segment B in here. Inversion, so reverse case, and translocation. Translocation in here. From another chromosome jump to another chromosome. And duplication. What is duplication? Here, duplication. Number 2. So why this one? Why this one makes sense? Why is it this one is make a difference in the protein? Makes sense is I mean in the protein expression or regulation of the cell system may be changed. Why this one is make different? Because simply is the segment it may be very big, right? And the gene inside them is quite small. Gene inside them quite small. So if this gene is they have enough the promoter or regulation factor enough for them, and jump to another location in genome. So that gene also expresses protein. That gene also expresses protein, right? Maybe no problem with our phenotype, no impact, because that gene maybe also they express well, and the number of amounts of the protein made from that gene also no change. So we have no disease at all. But the thing is not simple like that. Some case like that. Some case in the we can see in the DNA code we have that kind of stuff, and they have no any kind of side effect. No, but most of the case they make the effect because you know is you have the gene set and a gene cassette. I mean, you have the promoter, you have the start codon, and you have the stop codon. Anything is perfect. But the problem is we have also enhancer, and we have the repressor or silencer. They far away from the gene body. So if that one is also, if they break and just gene body jump to another point, and that means that gene is expression will be abnormal. It's not normal anymore. Okay, or somehow is a promoter break, or somehow is very important factor is not normal. And also remember, we have the in the chromosome. Remember, in the cells class, you have your chromosome, right? And some part of chromosome is condensed, very condensed, and that many gene in there is not expressed. And some part is of chromosome, they are decondensed. That means all of many genes in the decondensed region, they express protein. If the gene in the decondensed jump to the condensed region, I mean that gene is no expression. So that is simply explain for you for impact of the chromosome mutation. They have a lot of impact, and right now a lot of companies try to identify their chromosome mutation. I mean chromosome segment mutation, big scale chromosome, because of the very hard to detect that kind of case. I know it. Now scientists try to find out that kind of thing. Right now, the scientists can identify mutation. I mean, the small scale mutation, maybe less than tens or less than some hundreds base pair mutation, they can detect very well. But large scale, they cannot, because the technology is not really involved in that case. Somebody maybe learn from molecular diagnosis class. So you say that you can use PCR for detection, but that is a very few case we know about our genome. That case is already very well characterized. Very well characterized, so we can design a primer on that. And in many cases we cannot. Okay. Okay. And here is one very important concept, is a non-disjunction. What's non-disjunction thing? So this term will make their aneuploid. So the different number in the pair of the specific chromosome, pair of chromosome. For example, in this case, so the failure in the chromosome to separate during meiosis. So this order, we had a Klinefelter. In the sex chromosome, we had a three chromosome. In Down syndrome, we had 321. And Turner syndrome, we had only single X chromosome. So you can see that we have the sex chromosome. We have the three chromosome in the in the pair of 21. We have three chromosome in the also we have here 13. Sorry, right, 13 and 80. This case also we have the three. This is very specific, right? 13, 18, and 21 pair. Pair number 21, number 13, and 18, they have three. But we accept the X chromosome, accept the sex chromosome. They have the reduce or increase in this case or this case. You see it. Never we have another types of the chromosome. I mean, how about chromosome 6 or chromosome 1 or chromosome 10 relate with this case? Because of another chromosome, we didn't say that, yeah, right? But maybe they also happen, but they cannot come out to the light because maybe chromosome 1 or chromosome 6 too much important. If the people have three chromosomes in the chromosome 1, think about the impact to the cells. So that cell is not normal and cannot become the conceptus and cannot come to the real life, right? And how about the X chromosome? So X chromosome, remember, one X chromosome is a bar-body, so they completely one chromosome inhibit. So we don't need that DNA. We don't need the double amounts of X chromosome. So we lost one is okay, right? But if we only have Y chromosome, for example, we never have Y chromosome in here, right? Because if we lost X chromosome, we will die like that. That's simple. And this is also explained for you why they become three but not become one. Because if you are here is more better than less, right? This is dose of the protein maybe increase, but that is okay. Maybe abnormal, but still okay. But if we less, I mean, we will lose a lot. We will die. So the mechanism to make that kind of thing is unbalanced in the chromosome separation. Like we have the gamete with a two chromosome and one gamete we have any chromosome, and that is when we put this one and fertilize with this one, we have the trisomy, right? So we have a gene mutation. It's quite very simple and go fast. This one chain in nucleotide sequence in a gene, But this may be not very correct right now, because right now the scientists more and more identify new factors related with the cell activity. I mean, most of the information and most of the a lot of the genome reason they coding for regulation factor, not only the protein thing. The protein thing is right now we almost know about all the protein, but the regulation factor, maybe siRNA, some kind of RNA to help the cells regulate the cell activity, so we still missing. We don't know much information on that. So a gene maybe is not very correct. Think about is related with the enhancer of the gene, far away from the gene, maybe from very distant space. And first, we maybe this very basic, but we focus on the gene body. So we had a nucleotide substitution and a frame shift mutation and also gene duplication. And about the nucleotide substitution, we have a silent substitution, or we call another term is synonymous. I mean, it's no change in amino acids. So that's also similar with the I confirmed in the first slide. I mean, we have a lot of mutation, but just few mutation have impact. And missense mutation is second. We have a change in amino acids, but chain amino acid not really. We have the not always we have chain amino acids is have impact because of that amino acid maybe is far away from the active site, right? Maybe just in the tail of the protein and not function too much in the protein function. So maybe even we had an amino acid chain, but they have no any function. And nonsense mutation, that's a really big problem. They introduce to the stop codon to the RNA. So the protein is maybe stopped, but also due to the position of the stop codon. If the position of stop codon is a very end of the end of the gene, I mean, so the tail of the protein maybe it's not many impacts, so that is okay. But in the beginning or middle, maybe change. So just illustration for that one. So there one thing I want to check, because of when you sequence one DNA, you need to do analyze, right? And you need to find the start codon, maybe the ATG around here, and from that you can visualize your protein coding. But if you see this is a codon, right? So if the codon is they change in the nucleotide number 3, maybe your protein not change, and this is a synonymous a mutation. Protein not change. But if the mutation occur in the nucleotide number 1 and number 2, especially in number 1, in the codon, maybe you have the surely you will have the DNA, the protein, the amino acid coding chains. I want to mention something related with this one. Okay, turn later. And prime shift mutation is also the same meaning with the stop codon introduction to the DNA code, because of the prime shift made by deletion or insertion. They will make in that point the protein coding of your protein will be completely changed. And it also depends on the location of the mutation. And oh my, impact on the mutation location. So we had the promoter, we had an enhancer, we had a silencer. That is a regulatory sequence related with the gene. Because of in the eukaryote, this kind of thing is really complex. So we need to identify, because of when you try to identify because when you sequence one gene, you will see a lot of mutation, and you don't know is a message, and you don't know that mutation is really have impact or not. So you have to map into and you have to have the information. That position is what is the function of that DNA segment to the gene. That's maybe the promoter, that is maybe the gene body, that means maybe the enhancer. And another also very important is splice site mutation. Splice site is related with the intron removed out of the primary RNA to become the mature RNA. Remember, right? So splice site mutation will make that process in eukaryote, removing intron, not happen normal, and maybe the protein is completely changed. So we talk all of the scenario. The mutation can have impact to the protein amounts and also protein structure. Okay, we have in here is I want to introduce some technology related with checking the promoter mutation. Have any how can we check the promoter mutation? That is also related with my my project before. So I do in the gene is TNF. That is a very important gene, TNF. You see that here, TNF gene. So that this is briefly stand for tumor necrosis factor. That's a very important gene related with our immune system. So this gene, this protein, TNF protein, will be make the cell react with some kind of infection or some injury in the cells, and make the cell make some response. But the amounts of the TNF protein will have impact to the immune system occur in the cells, in body, in that kind of size, maybe the size of infection or injury. But when I checked the TNF between many people, and I found that we have a lot of mutation in the TNF. We have a lot of mutation in the TNF. maybe I have the one group of disease and one group of normal with that disease. And then I check the TNF mutation. We also have another gene, but I select that gene because of very good candidate, because of relate with the symptom of the disease. So I use TNF. So I can see that I had a question. Maybe the mutation in the promoter region make that TNF amount is a change, and that change will be impact to the disease outcome. And I just simply do this experiment. That is we call it the Lucifer assays method. So I just using some technology in the cloning. I just cut a PCR out. I cut the TNF promoter from two people, and I put into the plasmid. And that is the reporter gene, luciferase enzyme. So this is the promoter. We had two versions of promoter, right? We had the promoter of the diseased people and the healthy people. And I had two types of plasmid. And then I transfer into the animal cells. And then enzyme, you see that is the enzyme will cut the substrate. And the substrate will, after cut, they make the light. That light is signal. If more light, I see is more Lucifer enzyme. And that means more is show that promoter is stronger or weaker than normal, right? So using that kind of text, I can check this. My mutation is really have impact on that. Or I can do some good thing like I select normal people DNA promoter DNA, normal. And I see that kind of mutation. And I see that mutation may be important because it belongs to promoter. So that I use some technique in the cloning. So I change that kind of the nucleic acids, change only that one. And if the signal is changed, and actually it changed, my experiment is success, and I can confirm that mutation is important for the disease, right? For the disease. So done with that. Okay. So this is explained for enhancer mutation. So you see that enhancer very far away from the real promoter, even the promoter. And this is gene body. Promoter is nearby the gene. Maybe it's just upstream about some 100 base pair, or maximum is about 2,000 base pair. But an enhancer is a different story. Its enhancer may be far away from the gene, and this enhancer may be enhanced for another gene. One enhancer can use for several genes. I'm not sure about that. We can have a question. And the question is how scientists can detect the enhancer reason, because not very far from the gene body. So how can a scientist can detect enhancer? So one of the very popular method the scientists is use they just do the comparative genomics. That's one I teach before when I worked in professor in Korea. So you select you sequence all quite a big part of the genome, and you compare between the many people together, and you see that there's some part of genome they not belong to the gene body. I mean, it's not related with ADG or TAA something, but they far away from gene. But the mutation in that region is very less. The mutation in that very less. That means that mutation is not allowed to happen in that position. And I mean, that part is important to the cells or to the species. So based on that information, people can notice some important reason, because that reason is a mutation not allowed to be happen. You see, intron is have a lot of mutation because it's not important. They will be waiting for cutting out. And they have a lot of mutation, a lot of stop codon in the intron. But in the enhancer, no, very less mutation. So using that one, they can identify important regulation sequence in the genome. Okay, this is a illustrate for the splice size mutation. So the splice size is also simple story because you can use a DNA sequence you put into the internet software, and the software can calculate for you which one intron, which one exons. Maybe it's not correct, but somehow is they can accept in some level. And because of they have the pattern of the intron, here you can see that have the pattern of GT and AG in the 5' and the 3'. So what happened if the GT in here change to the AT? So that means this one is not, this intron will not cut. And the enzyme for cutting, I mean the spliceosome will cut another GT in the intron. And you see that a lot of DNA, a lot of the code DNA will be retained in the RNA, mature RNA. And they will have a lot of stop codon. And your gene is kind of no effective protein expression. This splice site mutation is a really impact. The same with the genetic frame shift mutation. So it's quite different with alternative splicing. Alternative splicing is a normal process in the cells. Using this one, the cell can make different types of RNA from the same DNA sequence. So that's normal for the cell activity. So, but the splice site mutation is quite very harmful because they make their they make the intron retention, and in many cases they not good effect. So next one relate with the thing relate with the mutation. Cause is a mobile element. So mobile element is first discovered by background McIntosh in corn. So you see that corn have a lot of color but it belongs to one tree. So we have the large element and the short element and very short element, and here is a more short element is tandem repeat DNA. This is quite in the course of the molecular diagnosis, I also mentioned this one a lot. So remember, in our genome, we have the 50% of our genome is a repeat sequence. That means that's part of DNA. We can find something same sequence in another region of the genome. And they have a lot of impact to our genome. Why? So, and this is one of very important and also very interesting retrotransposome. So maybe you all know about HIV, right? The retrovirus, very dangerous retrovirus. So retrovirus have a very interesting characteristic. And then they, for a long time ago, before we had a human, so retrotransposome already enter our genome and also enter another species genome. And they using some material in the cell to make RNA. From RNA, they coding for reverse transcriptase. Reverse transcriptase will make the DNA and turn back to the genome to make a new copy of the retrotransposome. So why is important? Because they freely jumping between any part of the genome, and when they jumping, they will destroy the construct of that genome, right? So destroy the gene body, destroy the enhancer, destroy the promoter, many. And also another very important impact, that is a retrotransposome will be provider homologous sequence. So remember homologous sequence, right? homologous sequence. So homologous sequence we will induce our chromosome with a chain. Well, remember the crossover of the chromosome. So without a homologous sequence, the chromosome is hard to change. So they need a homologous sequence, homologous site to change. So the retrotransposome right now or the repeat sequence will be have the important roles in the make the chromosome big scale mutation, like duplication, like reversion or deletion or duplicate. Yeah, and this is a type of the retrotransposome. You can find this one in genome of pig, mouse, and cow and cattle, many. Any kind of species have the retrotransposome in there. It means it's from our ancestor, virus already enter in there and they sleep in there. And sometimes if the mutation happen, if some mutation occur, I mean, so that's retrotransposome is sleep in our cells, DNA, but they sleep, they will wake sometime, and they make some somatic mutation, and they can make the cancer. That's why the virus can induce cancer in your body. And this is why it impacts on retrotransposome. So think about if the retrotransposome happen and jump somewhere in the cell. But think about the stage of the cell. If the state of the cell in there very early, like I mean, they happen in the cell mass, ICM, right? In the cell mass. So ICM, this whole body will be habit, right? Even the germline stem cell also have that mutation, and you transfer that mutation to the next generation. But your parent not have this mutation. That is one possibility. Another is late stage. You have the whole body, you have that mutation, and maybe you develop the disease, but your germline stem cell is safe. Maybe that means your baby have no any mutation for this, right? So somebody want to test parent mutation, maybe this is also one case you need to consider, right? And some kind of thing is they just have the mutation in germline cell, but whole body is normal. So your body is safe, but your baby have mutation. So in this case and this case, if you want to test that germline or the somatic mutation, and you want to test your parent, that means they are very confused, right? And so this is a mobile element to make they jump anywhere and make a lot of they destroy a lot of the CD in the genome. And a lot of disease related with this kind of mobile element, like with beta globulin, lung cancer. Okay, oh, no water here. Oh, we have a little slide. Okay, why don't we make a little break, about 5 minutes? 5 minute break. Okay, can I hear your voice? Can.
Okay.
Yeah. I need to find some water. So if you have any question about the slide or something, you can process.
Mm-mm.
Okay, so maybe it continues there. Thing because we have a lot of slide remain, and today in the end we have quite important stuff related with some the exercise we need to fast through. So okay. So we had more like here is some consequences of the mutation. I want to check again so we can you can hear my voice, right? I don't know, it's just someone well or not. Okay, good. Thank you. Meeting online is not funny because of the interaction is kind of very less. But that is the best we can do it. Molecular consequence mutation, that's listed you some case in here, but actually in the real world is more than that a lot. That just provide you in here some 3 cases that you can you can think about the possibility for another case too. So first is the gain of function. You have the kind of mutation here. Maybe the promoter reason. So number of the protein amounts will increase than normal. Normal here, allele1 and allele2. Allele2 have the alleles increase. So increase maybe is little good. And actually, abnormal is not good. We have to remove some thought about X-Men or Wolverine or something. Superman is not like this. So out of normal is not good for almost K. And here is the number of chromosome, number of protein expressed from one version of gene is increased. So compared with the last expression, that's good. In the second case, in B in here, so early one, we have just one case, and in here maybe the stop codon, so the protein making is not possible. So we can call this a log function is a haploid in sufficiency. That means it's amounts of protein, just 50% is not enough for a normal activity. I mean, it's a biochemical activity in the cells. It's not good. And they can make some side effect for the diseasing. But maybe they can make the baby. I mean, so the baby can can produce with this thing. Another one is dominant negative. What's dominant negative? Is we have allele1 is normal, and allele2 they have a mutation, maybe the amino acid chain. So this protein is produced, but the activity of the second protein is maybe inhibit the first protein. That means because the fighting between two protein and activity of the cell also is not normal. So this is several cases consequent of mutation. And that's a scientist only recall in this case. We have a vast case thing. I just read some kind of paper in last week related with mutation related cancer. We have many kind of new mechanism to make the mutation in the cells. I mean, we have the normal DNA, but somehow we have abnormal mRNA, and of course, consequently we have the abnormal protein also. Maybe from the some is I just learned from that kind of paper is we have some case like we have mRNA, but mRNA when they moving from where from the nucleus go out the nucleus pore, and the A nucleotide A and adenine will be changed to I. That means the inosine. What is this? I can write it for you. Inosine. So inosine, if somebody is maybe I talked to you before. I mentioned somebody already. So inosine is quite of the precursor of the adenine and guanine. So before making the guanine or adenine, so we have to make, our body need to make inosine. Inosine is not joined to DNA synthesis, but they join to in the pathway to make adenine and guanine. So inosine is quite important, and somehow the cell which have some factor will change from A to inosine. In biotechnology, if you join to the biotechnology, somehow you can somehow you have to use inosine in the primer design or some aptamer design. So that's kind of interesting. I tried already, and I had one paper published for inosine using. Okay. Next slide. We have the one example of the gene duplication related with gene duplication. That means amounts of protein making is increase, or somehow like case of the dominant negative decrease. So the disease is Charcot-Marie-Tooth. Oh, quite long. Maybe that's the first people got that disease. So they make the animal cells system in their synapse, not synapse, in the body. This body is myelin. Myelin. They have function boosting the signal inside of the cells when they go to the in the center of the neuron cell to the end to the synapse. So the myelin have the function for that. So somehow it's a myelin. They in the chromosome 17, that gene PMP22. If they have 3 copies, the amounts of the protein will be 150%. So 1 copy, 50% decrease. So both cases not good. So this is why they're not good. The phenotype we have disease form like this. So any kind of disease, so the scientists identify how about the prevalence of disease in the population. Maybe this one is we have 2,500 people. We have a lot of disease. This is the prevalence of disease outcome, not showing you the carrier. I mean, a lot of people have the whole. This may be the recessive disease. So recessive disease have just a lowercase a, and A have the phenotype expressed. But in many cases, we have the heterozygote, and this number don't show you in the population how many kind of people have the heterozygote, and they don't show up. But if they mating each other, the probability of the baby have the disease will increase, right? So another is when the tandem repeat DNA using this figure is many times. So this is a Huntington disease. Huntington disease is normally happen in the it's old people, normally in European. You know, it's the genetic disease in the Asia or Vietnam is very less. Not because of we have no disease. We have disease, but we have no money for the research. So Huntington disease is a disease happen in your brain. This is a normal brain, and this Huntington disease brain. So we have a big hole in here, and this phenotype people is not the action is not normal. They going is kind of not go straight like normal people. They go around and around, and somehow in the animal model, some scientists already create the very good model for the disease. They make the pig and they make the mouse, and Huntington disease make the mouse moving around and around in the room, but they have to rely on the word to go. They cannot moving independently. The disease made by the protein coding region of the CAG codon here. CAG codon coding for glutamine, the glutamines. So I mean, this is normal, and in this abnormal, the glutamine amino acids amount in the protein increase, and that's make the cells, neuron cells is degraded, and make the disease forming here. A lot of disease, right? Question? No question. Another one is, this one is very important, and in Vietnam we have a lot. So the disease related with the hemoglobin gene. So hemoglobin gene is we have 2 chromosomes, human, in the chromosome 11 and the chromosome 16. So this is a beta globulin cluster, and this is alpha globulin cluster, and some kind of gene is expressed. You know, it's function of globulin, right? This is a protein, mainly structure of the red blood cells to transfer the oxygen from our lung to in any part of our body. So if the mutation of that mutation happened to this gene, we make a lot of impact because of amounts of oxygen is not normal anymore. You know, we realize in oxygen, right? So in here, epsilon, gamma, and delta globulin relate with when we are concepts or when we are baby. So that gene expression, but when we are become a little bit mature, so beta globulin take part expressed, and this gene will be not expressed anymore. Alpha globulin also the same thing. Theta also expressed, and also alpha 1 and 2 expressed. So in the mutation of that gene, we will have a trouble with the oxygen amounts in the cells, any cells. So this is a consequence, kind of the disease related with that is thalassemia. In Vietnam, we have a lot, especially in the highland, of Vietnam, and also in the north of the Africa, or some part in the Green or Italy. They also have this kind of disease. Prevalence is increased. So the hemoglobin disorder is one of the very famous diseases, sickle cell disease. So we have these normal cells, and if we have the mutation in the beta hemoglobin, and our cell will be like this. So the round cell is good for the cell for our body, because of the round cell, they can move inside the blood vein easily. And think about this kind of cells, they moving not freely, and they got stuck, and sometimes we have heart attack for this one. So very harmful for this is heterozygote case, and in some case of the homozygote, they very die soon. This belongs to missense mutation from valine to glutamic acid of the beta chain. Exactly in the lab. So many people in our class, they practice to detection, design a primer for detection thalassemia or no, detection of sickle cell disease. Because of that gene, hemoglobin gene is quite very important. So any mutation in this gene also relate with the disease. Some mutation make the sickle cell disease. Some mutation make the thalassemia disease due to the position of the mutation in the gene. And this is a kind of heavy thalassemia. Heavy. So we have a big problem with body development. Next, we go a little bit about the consequence of mutation. Now is we go to the cause of mutation. I'm mixing something. So remember, in any kind of duplication process, so the cell will reproduce. The cell will be introduce some mutation in the new DNA. Sister cell will be have a different genome. We have the mutation rate later. But in here we have some induced mutation by mutation. That means that some new chemical could be induce the mutation in our DNA. Remember the very toxic chemical in the Vietnam war, right? The chemical is I check. Orange agent. So the U.S. Army using a lot in the Vietnam War to kill their jungle, to remove the jungle tree. But in fact, and make a lot of the soldiers from Vietnam and also from U.S. side had the mutation, and they got cancer. They got a lot of the problem with their health, and they got maybe you can see that a lot of baby, the next generation of the soldiers, they got the mutation and defective a lot. I mean, the phenotype is abnormal disease form. That is a orange agent is a quite a very famous mutation. So we have that one is base analog. So that's base analog is we have ADCG, and that's some chemical. They have the structure very similar with the ADCG. For example, this 5-bromouracil, and they can when the DNA they make a new strand. This chemical can join inside of the strand, and DNA polymerase cannot distinguish them, and they make a mutation. And nitrogen mucin is the most dangerous here, and we had a formaldehyde in the agriculture in Vietnam. Many companies using formaldehyde, and or in the lab we also using this one. The toxicity kind of medium, not very high, and aflatoxin B1. So aflatoxin. So the chemical when we eat something is have the mucin there. So they make aflatoxin. Normally our liver is affected. This one. And another one is the virus. I explained already the virus when it jump anywhere in our genome. And also is we have something related with physics like radiation, like we have X-ray or a nuclear bomb in Japan also, and X-ray. X-ray also harmful. Don't take the X-ray picture a lot. Or if you hurt here. And we have another factor is non-ionizing. Ionizing is they kind of high power, so they break the water. So that is becomes free agent moving around the cells and make they track some structure of the cell. So we have UV. UV light also very toxic. So like I read in some paper, just 2 minutes. Less than 2 minutes, your DNA will be destroyed. 2 minutes, if you have intact enough, the DNA of bacteria or the cells will be destroyed by UV. So some part of our planet, like Australia, the UV concentration, UV amounts is very large. So the Australia people is normally suffer with UV light. They have a lot of disease related with skin, maybe the tumor development for skin. So one of the people also ask why UV have what effect like that. So they found that they make the pyrimidine dimer. For example, UV light very high power, they can make the DNA like TNT pyrimidine will be remove this part. Two kind of T will be connect together than normal, and that position will be break. And when the DNA make the fixing, the fixing is not correct. Because of many kind of mechanism related with DNA mutation. So in the cell, from very long times ago, the cell already developed the mechanism for fixing or repair DNA wrong coded DNA. So we have many gene related with this system. So that gene also is it's a good thing. We have a lot of the protector in our genome, right? We have a lot of the detector detect and fixing for our cells. But if we have any problem with this good gene, I mean, if the good gene is die because of mutation, our cell is really have a problem. For example, in here, very famous cases, we have BRCA1 and BRCA2 gene. So this is, if we had mutation in that gene, so we will have the very high, quite a high risk of the cancer. Yeah, the cancer for another gene also very important. And this is a upside is the mechanism of the gene action. And then we're talking about the mutation rates. In here, one base pair have the risk of this this race for one generation after preparing. That means a 10 million base pair, we will have one base pair mutation. And think about our gem have the 3 billion base pair. So any kind of the application, we have the 30 new mutation, right? 30 new mutation. But think we have just 30 new mutation, but where they locate, mostly they jump into non-coded DNA because of the 98% of our DNA is a non-coding DNA. Does not relate with any exons, I mean exon. So they have no any function. And also this happened with the gene size effect. So we have a quite, in gene size, we have very diverse. So some gene is quite very short, maybe just 900 base pair, we have one gene. That case relate with the olfactory receptor gene. The gene coding for the receptor in our smell neuron cells, smelling, making making ability for our can smell. And that gene very short and it don't have the intron in there. The size about just less than 1,000 base pair, very small. So thinking about we have a 30 base pair. So the new mutation will be probability of that mutation jump into the smaller size of genes with a small size is not possible. Some genes are very big and they suffer by the new mutation, like the new rock from the universe come to our planet, join to China or the US first. And Vietnam is so small and normally we don't have the rock from the universe, sorry. And we also have a hotspot. So the hotspot is the place of our genome. Normally they have the higher probabilities of the mutation compared with another part. So we have the maybe CPC site. This also very important source of the mutation. Next slide we will see that. And another one is the ACE. When we get old, our genome copy system is not perfect, and they make more and more mutation. For example, in here, so that syndrome is several times higher for a father as older than 50 years old, compared with a father in 20. So if you get married and you had a baby in very old, that also the risk of the baby will be have the mutation. Okay, next slide will be explained for the CPG size mutation. We had a cytosine here. So cytosine and thymine is very similar. But in the CPC side, so especially C and G. That is a mechanism related with the epigenetic. So we have that kind of methane in here. Adding is C. That C should be next to the G, because that is a recognized size of some specific enzyme in the cells. The cell will have some enzyme to not randomly. They have the very strictly add the methane to the 5C. And then this one is not stable. Not stable, and then quickly change to thymine by the amine out of the structure of the cytosine. And then now is from C we have T. So that we have mutation happen, right? And this is very popular. So many sites in our genome is have the methylation site in CPG. And this is A effect. So we see it's a thing about some syndrome, the disease, when the age of the age of the parent increase. And next is kind of easy graph there. Mutation detection. This is related with the course of the molecular diagnostic industry. Molecular industry is about a lot of money. A lot of companies join this one. Many companies, right? And this is related with application molecular diagnostic market. We have infectious disease. Remember the COVID, right? So this outbreak, so infection, that's the infection disease by virus. So we have many companies making the kit very quickly to make the kit, and that is make them a lot of revenue. And the blood screening. So anyway, when you come to the hospital, they take your blood first or urine, and the genetic testing for kind of germline mutation, and that germline mutation is good or not, and they can predict for you. And after this course matching, you are the one to calculate for the risk after got the result from genetic test and from germline mutation. And this one is oncology testing for the cancer, the tumor sequencing and detects for treatment, because you learn about pharmacogenomic, right? Some kind of gene is mutation, and using that drug is not kind of good. So we need to change to another drug using another pathway of the biochemical pathway. So we have a lot of methods. The number of methods is a lot, really. Okay, so this is just showing the percentage of the amount of money related with which kind of the division in the molecular diagnostic. Okay, we finished this lecture, this origin and detection of the variants. We have time, so now. Hello, can you still hear my voice? Hello?
Yes.
Okay, good. So I go to next way. Okay, I will go to the next slide, next presentation, because we have to go quick a little bit. So detection variants. We have a 30 slide, but I go a little fast. We can enough time, I think. So. Oh, this is not compatible. So do you have any question about the this recording, right? Any question for the first presentation? I go a little fast. If you can know it and write it, and we can discuss. I'm not sure I can answer all of them. I'm not sure I can answer all the questions, but we can discuss. Okay, if not, we can go to another one, also the same thing, related with the detection of the variants. And I will go fast on this one because of this one mainly talking about technology for detection of variants. But the end of all slide, we had very important, or quite important, we can use it for calculate the risk for the disease or some probability calculation. This is quite a basic for. for our course. How can we open this one? Detection of variance. Okay, so different with the meaning of the course of the molecular diagnostic, we use some technology to confirm that people have the mutation or not. That people have mutation or not, and based on the percent of mutation, we can make some conclusion that people is infected with the disease have the risk of the disease or the risk of the cancer or not, right? But that is already using information of the scientists already provide for us, because of people is the scientists already detection show it that mutation is quite important for disease development. But in here, thinking about bigger scale scenario, like you also using that's for detection, but you also detection variance. Okay, detection variance, and based on detection variance, you can make the research. You have to do the study to confirm that that kind of variances have impact or not to the disease outcome, or the benefit for the people or not. So we have several simple I summarize here for you, from the simple one to the complex one, from the very old one to the new one. Homework is now using, and you know, it's if you're running and you're seeking for the technology is not meaning, so you have to thinking about the meaning of the technology, how the scope and the price and the compatible with your system is also that's more important. And second slide is the very first sample of human variation. That's it, blood type. So for a long time ago, we kind of very interesting in the different between the people with people. That girl is so beautiful. That boy is so special, so smart. That is so tall. Is that one? Another one is so short, so fat or thin. And one of the very first from 19th century is the blood type, because of we know we can, when we lost the blood, we can transfuse the blood from another people. So the blood transfer is we depend on ABO blood group. That is the ABO system in the blood antigen is very famous. Anyone knows, and I think many people in the world have information about your ABO typing. And the second system is also very famous in the blood antigen system is our RH result system. That is two most famous. But in the blood type antigen system, we now, I checked, is that we have about 39 systems we can time for. But ABO and RH system is the most dominant and the most used system before blood transfer. So this will just go fast a little bit. Is ABO blood group discovered by Karl Landsteiner in very good years, first year of the 20th century. And this ABO blood. So we had a red blood, we have the type A, we have the A antigen in the red in the cell surface of the red blood cells, and type B we have only B genotype. So AA and AO different is amounts of the antigen in the red blood type, and O type is we don't have. Actually, we have it, but the structure kind of less than compared with A and B. And of course, in the type A people, we develop some antigen. Antigen is not bind to the A, but they bind to the B. This is anti-B antigen. No, sorry, antibody. And then the reverse case, we have the B type, we have the A antibody in the blood. and AB we don't have it. Of course, because of that is science of the antibody is the story about antibody making. So antibody should be make if any cells, is antibody making by the B cell, right? So any B cell is, of course, from the beginning, any kind of B cell will be produce antibody to bind A. But any kind of antibody bind to A, our body will be removed out of our body. They will kill. So they just remain a B antibody. And always they have both. So this is simple, skip. And this is kind of structure of IBO blood group. So we have this is structure of A and the structure of B. So CSO also have that kind of the antigen in the cell surface. This is glycoprotein, the sugar. You can see it is fructose, lactose, and glucosamine group inside in the cell surface. Glycoprotein. So glycoprotein from O, they have 4 group, and in here B and A they have 5 group. So in here we have more lactose in B's, and in the A we have the acetylene glycosamine. So just small part of the sugar. So our body is making antibody really effective for small part of the antigen. So this is very famous. You use antibody for typing your blood using aggressive reaction in the blood. So this is control. This anti-A will be react with the A. That means this one. So this one's simple, and so I skip it. Resource system is another protein in the red blood cell also. So I go to part of the method for detect the mutation. So first is the protein electrophoresis. So the same with the DNA electrophoresis. Anyone is familiar with DNA electrophoresis. DNA is have the protein also the same thing. But the thing is, what is different between the protein and DNA? So DNA is they have the negative charge in the DNA strand, but the protein is not. Protein is we have the 20 or 21. Actually, we have the 21 amino acids, not 20. And any kind of amino acids, they have their different charge. No charge, positive charge, negative charge. So they can diverse. So first, we have to put the protein become their all negative charge using the SDS. SDS will provide negative charge for the protein, and then we can run from the negative charge to the positive charge like this through the gel. So normally we have to use the 5 gel not by agarose. We also okay, but agarose is a quite big holes, and we have to use polyacrylamide gel, the smaller gel, have the better separation of the protein fragment. So this is protein. So the principle of the protein electrophoresis is in the protein chains. I mean, some mixed sense substitution. We make the protein amino acids protein. Any single amino acid chain will make the chart of the protein will be changed. So that means the chart changes, and then the protein moving will be different with the native one. So in here is sample. In the sickle cell disease, they run in the protein for beta globulin gene, and they can detect here. So this one is a sickle cell disease. This is a normal, and this is a heterozygote of the people who like carrier for the disease. You use the protein, you can detect that one. But a protein experiment really take time and quite expensive, and not very expensive, but they take a long day for setup. I don't like it too. Okay, and also is we have a very limitation is very limitation of variant can be detected for protein electrophoresis. Okay. Next is we use a silent blot. Silent blot. So we cut. The principle is we cut our DNA by specific enzyme, and for example, A alleles and B alleles they different in the resistance of enzyme site. So in here we have no cut, and this one we have cut. And if we have no cut, we have a bigger band, and if we have cut, we have a two smaller band. And using this one, we can distinguish between the mutation or not. This is silent blot. So because of the why is silent blot? Silent blot because of we have to use some prop. Prop is a kind of the fragment of DNA with the fluorescent, the color. So think about we have a genome 3 billion base pair. If we cut it, they have a lot of band like this, and it makes you confused when you want to type it. You hard to see that. So they have several methods and transfer, and then they ligate with a prop. The prop is designed specific for some part of the DNA, and you focus on. And you remove all of non-specific prop. You just focus on some prop in here, and based on the pattern between the land, you can see is we have a difference in DNA coding or not. Next one is also a same limitation with protein electrophoresis. Is almost disease causing mutation do not affect by restricting some enzyme site. Very and also this method is reproducible ability is very less. I mean, this time you generate this pattern, the picture, but another time you cannot make that picture again. That is a very big problem if some scientists want to confirm that one in his or her lab. Another is a tandem repeat polymorphism, and this is a method is many lab using right now. If we have time, we will more detail about this one. But I believe a lot of you join the class of the molecular diagnostic. We have basic this one very good. So the principle of this one is allele A is have the 1, 2, 3, 4 copy of this repeat. Very short repeat and neck together. And B alleles, we have the 1, 2, 3, 4, 5, 6, 7 A allele repeat. So if I design a primer in here, universal primer for forward in here and universal reverse in here and here. So the primer will bind in here and here, both. So they will generate a different size PCR band, right? So based on the band size, you can tell that two people is different or not, or that people is a heterozygote or homozygote of disease form or the normal form. But in this case, this kind of method, you have to use a very exact, this is a DNA electrophoresis, but you cannot using the simple apparatus for DNA electrophoresis. You have to use a very good machine for very high quality and high resolution in DNA electrophoresis for this method. And some case you need to use, you have to use a color. I mean, the probe or the dye to detect the signal. You cannot detect by your eyes. And next one is we talk about SMP, single nucleotide polymorphism, and copy number variation. Sorry for I didn't put the full name of this one, but if you start using this this term, you can easily get it from internet as well. So this is the first method we already confirmed using the enzyme cut to see the pattern. And this is a SCM method like this one. And this is CNV. So the repeat sequence is more than 100 base pair. Sorry, 1000 base pair. This one and this one is similar compared with these people, this chromosome. So maybe this one is contain one gene in here, one gene in here. So the double case of the double amounts of the protein here. So NNBs we talk about just the single nucleotide change. So in here C chain to T, and this is a complementary TNA CNG complementary. From the large scale to small scale, CNB and SNP. So PCR, so anyone is, I believe all of you is remember what is PCR. And this is, I just mentioned that we have a lot of type of PCR reaction. Thank for Terry Mullis, so who died last year. So he first people got Nobel Prize for making a PCR method. And another scientist is trying to modify PCR. We had a multiplex PCR. I mean, you can detect the several segment in the DNA aggregation, and nested PCR or semi-nested PCR, touchdown PCR to increase the specificity of PCR or AIMS PCR. So you practice in the lab using this method. And in qPCR, by real-time PCR, the method for detection the COVID virus recently, real-time PCR, gold standard for detected virus right now in the world, or high-resolution melting method PCR. Okay, next, we have a DNA sequencing. But it takes time to explain this one. So you use electrophoresis and the DNA synthesis using the dye. You can run in a machine to get a pic like this. And this is a good. This one is a very good picture. So we had a pic like this, and made it on the pic. The computer for the computer will be calling for the base calling for you CG like this. And but the thing is, the life is not easy like that. Sometimes you got this pattern. This is my result before, because I normally running the sequencing machine, and I got a lot this one. So this is also same with this one, very high quality, but you got heterozygote, a lot of case in here. This is homozygote, there's hetero in here, a lot of hetero. The thing is, the problem happens. So you don't know is which base pair belong to which chromosome and which one belong to what chromosome. I will explain this one more clear in the class. I think I need to roll something to help you to grab this one. And high throughput DNA sequencing NGS, very good technology. This is a game-changing technology. So $1 billion US dollar for whole genome sequencing from 20 years ago. But right now we can decrease the price to the 1,000 or 2,000 US dollar for one genome. Very good. And you can check for any part of your genome, any mutation, and predict for any mutation you need to focus. Maybe several gene, maybe 100 gene, or whole genome, due to the money you have. Yeah, this way the thing to analyze this. In here, they will make a lot of sequencing read for you, the sequencing information for you, and you see the red and here. In here you have this one look like this one is a heterozygote case, right? And the yellow one in here is a reference genome. After have a sequence, you have to compare with a reference genome in the internet. All the work need to compare with reference genome. And this mutation, the question is, this one is germline or somatic. Look to me, this is a germline mutation because of the A we have 1, 2, 3, 4, 5. And then G is a 1, 2, 3, 4, 5. 50 and 50. That means one chromosome has G and one chromosome has A. So that means it look like germline. So if the G is A is about 100 and G is about 20 or very less like that. So we can think about this may be the somatic mutation, right? And if you compare, and this is a first one to a large scale scene. And if you want to confirm, you can use a single sequencing. This one to confirm using another part of your DNA, like the first question to you to compare. This is a germline or somatic. Simple. Thing is, you have money or not. And another one is quite an old technology, but still used a lot. That is using DNA probe. And large scale, we have a DNA array. We still have time, right? We can do that. So DNA probe is the single strand DNA, maybe just about 15 base pair, and they have the dye in the tail of the DNA. And when they bind by complementary to the probe, the probe is already in the place already. The probe in here, and then I mix my DNA in here. So if my DNA have mutation, we have the red color, and if we don't have the mutation, we have the green color. And if we have the heterozygote, we have the yellow color. Sorry, so not like that. So if we have no any kind of mutation, we have the green color. Based on the amounts of the dye, we can check that people is heterozygote or homozygote. So this is summarized of the DNA mutation detection method. So require a lot. Methods is real a lot, and many more thousands of methods every month, every year making. So you just think about using the specific and best method is suitable for your lab and suitable for your system. Yeah, like the method for checking the COVID-19 in Vietnam, we also had a lot of debate. Yeah, a lot of debate. If you learn deeper about that, you can understand where, and no need to debate in the internet, in the Facebook, let's say. Okay, now we move to quite an important part. We have enough time for that. This is we can call it population genetic. Very important. So if you just focus on hello. Hello, Nam. Talk to me.
Yes.
Okay.
Yes.
Thank you. Thank you. So if you want to just focus on using the small amounts of people, small number of people, you just stop in the diagnosed, right? You have no any no any kind of you have to just talking about diagnosis, that people have a mutation or not, right? But you cannot answer the question, like the big question, big picture, like why North European is taller than Asia, like here, and some question I know in here. So why sickle cell disease is common in Black America, but not in Asia? And why cystic fibrosis is much common in European compared to Asia? I mean, it's a different population have a different phenotype. That phenotype not only the tone, the skin color, and also the disease, right? So you have to learn about the population genetic. That is very important. And using the knowledge about the population genetic, you can answer the question. And if you want more deeper, you need some some kind of method for confirm, like using the cell technology, using the molecular technology or cell technology, or even using in vivo technology for confirm using that mutation. But right now you move a little bit medical genetic really close with the population genetic. So see the gene or genotype or haplotype frequency. We're talking about the talk very much about the biostatistics related with genetic statistics. So I go deeper a little bit. Where is it? Here is first is a basic concept in the probability. So first thing I want to mention is independent event. This one is you learn in high school already. So independent based on this one, you can have the rule of the probability for the rule of the multiplication or addition rule. Quy tắc cộng và quy tắc nhân. Of the calculate for the probability using the cells. But you have to consider very important concern when you recognize that kind of term, that when you want to apply the rule, which one, multi or addition rule for the your eye cells. For example, independent, like I want to have a first child, I want to have the second child, a third child, a fourth child. Thinking about the event, they have the relationship in the biology effect or not, right? So first child have no any kind of relationship with the second child, right? For example, in case of the sex control is illegal, is not allowed. So the first event is the first child, maybe the sex of first child and the sex of second child may not relate each other. This is independent event. So multiplication rule is if you have to do the work, but you have to several steps, and that step is have to be done. Maybe you have 3 steps, and you have to be done that step. So you need to apply the rule of multiplication. And addition rule is if you don't care about, you have to finish the first, second, and the third, and they not relate each other. So now is and, chain, by, or. So you may apply the addition rule. So I put the example in here. So what is the probability of the one couple have 3 daughters, right? So the couple need to make the first child is the girl, second child is the girl, and third child is also the girl. They have to. So the P will be 50% for the first child because of in the biology we know this the sperm. I mean, it's the male sperm and female sperm is similar, right? So 50% of female sperm is cause for we have here is first child is this one, and the second child and the third child, and because of the event is they need to kind of they have to be they have to fast the child first. So we using the rule of the multiplication. So now is the P of the have 3 girl is a 1 for A. Our addition rule. So what is the probability of one couple want to have 2 child with both sex? Maybe 1 boy and 1 girl, or 1 girl and 1 boy. That's not important about the order, the sex of the both child. So boy, girl, or girl, boy. So all in here, so we apply for the addition rule. So first we have to making the probability for having a child with the same sex. So P for the 2 boy and P for the 2 girl, like this. And simple, right? And And this is a P of the two tri have the same sex, so 50%. So P for both sex will be 1 minus 4, 0.5. So we have a 1, 2. Oh. Next, gene and the genotype frequency, just remind you this part. So for example, I need to type in I have the kind of research, I have the money and I have the grant, and I need to investigate people with very important genotype gene. That gene maybe had a big impact for the disease outcome of that village or this town. So that gene have two alleles like M and N. So genotype result will be 64 people have MM and heterozygote 120 and NN is 16. And the gene frequencies is, from each allele is very easy to detect in here. So the frequency is this number. Very easy to calculate, right? Don't need to use any software to do that. And this is Hardy-Weinberg principle. Just remind you, I think. So, but you have to admit that this rule just apply for the case of a random mating. So, so A allele in the frequency in the population we have the frequency is P and small a we have a Q. So this is a distribution of the probability of the heterozygote and homozygote in the uppercase and homozygote in the lowercase. Yeah. And in the real application of Hardy-Weinberg is look like is a very ideal situation. No any kind of population have this one like random melting. The random melting is not like that. We have to another factor will affect this one. But this is the principle. That is in the perfect circumstance, perfect scenario. But in the real research, in the real study, you also need to use this one. And then I also use in my paper and my publication. So you also need to calculate for the A alleles, small a uppercase, and the frequency of each alleles. And based on that, you calculate for the Hardy-Weinberg principle. But if your calculation, I mean, it's incorrect. But if your calculation result is kind of very different from the Hardy-Weinberg principle, so you have the conclusion, this one, this population is not random melting, it's a one conclusion in the genetic. So this is very important. Maybe it's simple, but very important in the genetic, and also using for our course a lot. But that difference it should be significant. I mean, some p-value should be less than 0.05%. And this one is we're using the Hardy-Weinberg rule to estimate for the frequency of mutation carrier. How to make this one is more compact to you. For example, your friend. For example, using this example. So you have one friend. You have one friend, and they ask you that. She asked you. She had a boyfriend. And her boyfriend is in the village, and there one research that village have quite a high frequencies of the diseased people. We not care about what kind of disease, but diseased people, and the frequencies of that disease is quite high. And this her quite of she kind of very worried, quite worried because she loved him a lot, and she would have baby with him after marriage, and she ask you about the possibility of the baby is have the mutation or not, and ask the question like is she have the mutation, that mutation in her gene or not, secretly. So that is quite a question, right? And you need to answer. Secret answer. So this is one example for that. So we know in European that one disease like cystic fibrosis is very harmful for people. And the prevalence of that disease is 1 per 2,500 people have one people have the disease. And disease. Remember is remember that this one is a recessive disease, right? And this number is sorry. In the Hardy-Wender calculation, this is a AA thing, because only the homozygote of the mutation can make the disease form, right? This is this one. So you have to make the frequencies of this one. You have to square root of this one, and you got the sorry, this one calculation easy, right? So the frequencies of the mutation, and this is normal. So that is a normal is you simply like 1 minus for this one, and this one is very close to 1. And then frequency of carrier is about here. So 2 minus this one and this one. So this number is quite interesting. That means in that village or this population, each 25 people, there are 1 people bring their mutation. That means in their in the population. So if the boy, if the girl want to get married with the boy in there, so the prevalence of she got the husband and the mutation is how much? How much? It's a 1 per 50, right? Because we have this one is all of human. So, but we select only for boy. So this number will be decreased a little bit, right? So now is based on this one, you can see is we have calculate for the risk of disease or calculate for the disease carrier prevalent. And based on that, you can suggest some strategy to eliminate that harmful mutation in the population. That's the work of the genetic counselor or geneticist in the future. Okay, that's the meaning of the course, right? So next is we have 2 more slides. We just briefly talk about the variation frequency effect by some factor. You know, its mutation is only happen by nature, and sometimes by the mutation. But without a mutation, we just focus on the nature mutation. And nature mutation, when they making, if the fetus can develop to the full term and making the fully development human, so they will adjust first with the nature selection. Nature selection, we talk about the environment around the people. And if that is a good thing, the nature selection will be favor that mutation, will be more and more in the next generation. If that's not good, they will eliminate that. And for example, very much study focus on the reproductive trait. So if this mutation happen and that species or that human have a lot of baby, so a lot of baby is that means that my genetic transfer to a lot to the next generation. That's good for biology of that species or that stability. Think about in Europe for a long time ago, many countries or many part of the village or the part of the Europe, they are just some very small people, small family can make the whole population right now. For example, in US, I visit Utah states one time, and there is we have just several family. They come to Utah, and now they make the big society in there. So you see that we have a lot of the mating with a close relationship in the biology, and a lot of diseases we outcome and recognize in the phenotype. An interesting part related with sickle cell disease and malaria. So sickle cell disease, we're thinking they have sickle cell form. They help the people with this genotype confer better resistance to the malaria. So you see that here the picture is a distribution of malaria in the Africa, and also in another country like Italy, Green, and this one is what? Turkey, right? Turkey.
Portugal.
What? Saigon? No, Saigon.
Portugal. Bồ Đào Nha.
Really? In here? Okay, we check it later. Oh, right.
Yeah.
Okay, so this malaria in here, so this is the frequency of sickle cell gene. So they confirm and they say that sickle cell disease really eliminate the development of malaria in the cells. So that is also good for African people. But when African people moved into the U.S., you see in U.S. have a lot of like people, right? There's also African moving on, and they the sickle cell disease right now they don't have provider good thing, and they now become a bad thing because in USA they have not a lot of malaria or mosquito, so that's don't need to become harmful. And also we have a case interesting. It is lactase persistent. You know, it's when we are baby, we have enzyme in lactase can digest the milk from mom, right? And but when mom stop milking you, and then the lactase is also decreased because of that's the biology and the evolution. You don't need lactase anymore, and that's gene stop. But in some population in the world, especially in some country of the Africa, and also some country of the North European, like Finland or Netherlands. So the people in there still using the cow milk, right? But in Asia, we don't use cow milk for a long time ago. Recently, we use that by Vietnamese. That's people from this population will be developed. The lactate will be still expression because of they use the lactose sugar still a lot put in their stomach, and the gene is expressed. So that's it make very sense for the variation frequency. That variant is in Africa and also in Europe. I also thinking about the thalassemia in Asian people. Thalassemia also that's my idea. I don't know if somebody did or not. So thalassemia is a disease related with the beta globulin gene, right? Beta globulin gene, and they also this country also suffer with this one, similar with malaria. And think about the Asian people, Vietnam, Thailand, Malaysia, or Indonesia, they also suffer with the thalassemia. And there is a relationship with the thalassemia and a mosquito relay on that. Thalassemia and the mosquito. And another factor related with the variant frequency of the genetic variants is genetic drift, like the unbalance of the probability. Somehow in the mating and make the next generation, some alleles is a favor. So randomly, this allele because of just random, no any kind of reason, especially in a small population, and they affect very much by the founder. I mean, the very first ancestor, very few ancestors in that population. So, and then genetic drift can increase or decrease frequency of the disease. Like in Finland, we think about a very small number of ancestral people long time ago, maybe 1 or 200, 200 generation. This gene is will remove our Finland. Maybe that's first people they don't have the static fibrosis and they don't generate this one. Or this is why the decreased frequency of disease. And they say disease is common in these people in the Belgium population. So they increase disease people. And also we have the gene flows. That means these people with this genetic of this population jump to another population and introduce a new mutation to a new population. Or lethal mutation, that means that mutation is they cannot develop to the full term and no baby can come out. Or somehow that people is infertile, cannot make generation. And very important is combined effect. Okay, we kind of finished the class today, so also finish the time, kind of extend a little bit. Why don't we have the report, the shirt? I need the report a little bit because we need some score somehow. Okay, are you there?
Yes.
Okay, I make the report, the thing related with the report. You may have the shirt or at least discuss about because I don't know this one. I'm also curious about this one. About the thalassemia and malaria relationship. I will make I will announce the report into the Facebook, and you may make it and submit it in the Blackboard. Okay. So any question or okay, thank you. Okay, we can start now. Thank you very much for joining today. Surely, I will upload the slide, any slide I make it, I upload it. Good. What else? Okay, you can stop and enjoy the day. Thank you.
Okay.
Bye-bye.
Goodbye.
Now to stop this one.